to join, but it seems we would begin while others join. And without further ado, I would like to jump straight into the presentation because we have so much to discuss. Welcome once again, and today we'll be discussing about something that is very pertinent and very close to us in Africa as regards what bioinformatics and what Africa stands to gain from bioinformatics. So, and today we'll be talking about something very serious: predicting health crisis with genomic data. How to predict health crisis with genomic data, and how Africa and the rest of the world can leverage modern bioinformatic tools, resilient infrastructures, genetic insight for predictive healthcare. So basically, we we are intent on using bioinformatics for predicting health crisis. So the world has moved from reactionary but preventionary. So, and how does bioinformatics come to play? And how can Africa leverage this bioinformatics and especially the world at large? So, I remain my humble self, Dr. Ashraf Akintola, and this is a webinar organized by NOBLEKINMAT Limited. The first part of this presentation, I would talk about the pathogen genomics revolution and the fact that the world is globally, generally shifting uh, uh, from reactive emergency containment to proactive predictive genomic intelligence. So it's just like we had it in during COVID-19, where we had a lot of emergencies globally. And even though it was predicted that Africa will bear the biggest brunt of the pandemic, but for God so loved us so much that we never had as much casualty as even part of the developed world. So hundreds of thousands of people died in Brazil, Ecuador, America, Canada, Spain, Italy, name it, France. So unlike in Africa, in fact, it was as if nothing happened. I was when the whole thing was unraveling, uh, Daegu was the epicenter of this COVID-19, and I was in Daegu. We were in Daegu. My place is Daegu. So from Wuhan, the first epicenter in China. So we had someone that traveled from China to Korea and then Daegu. So Daegu became the second most popular epicenter of COVID-19. And it was serious that time. So everybody was indoors, and later on when we were not indoors, using mask and other preventive measures was kind of compulsory. In fact, it was very tough that time, but we thank God. It was so severe that if someone in your close contact has COVID-19, like they did a survey, they did they sequence, they took someone and that person has the sample of that person has COVID-19. What they are going to do is that everybody that came in contact with that particular individual must do 2 week quarantine. So, and the use of a private personal uh, um, COVID-19 detection tools was also common then. And it was very rampant that if someone in your lab or your building has COVID-19 and it's covered, everybody is affected. So that's how severe it was. Eventually, I also got COVID-19. I also had COVID-19 at that time. And I was so happy. It's kind of counterintuitive. How would you have COVID-19 and be happy? So because I know that's mandatory 2 weeks holiday, so I went home and nothing happened. Yeah, something happened. I was feeling headache and it was just mild, but I knew I had COVID-19 and I did the test and I did the test myself and then self quarantine with my family and that's all. So what I'm saying majorly is that the world has moved from that kind of reactive emergency containment to proactive, predictive genomic intelligence. We will predict it before that incident happened. And recently, there was a global pandemic that was supposed to be a pandemic. Sorry, there was something that happened. That was an outbreak that was supposed to be a pandemic, but it wasn't. It later didn't become a pandemic. So I think it was one. I forgot the name of the disease. There was a kind popular meme online that nobody even thought of this. How can you be so useless as this kind of disease? I've forgotten the name of the outbreak. So
Antivirus.
Did you talk
antivirus.
about antivirus?
Yes, yes, antivirus. So it was so funny that nobody even thought about it because yes, there is a more advanced surveillance and outlook and reaction and ways of managing such diseases. So unlike how it used to be, even Ebola that came um, that one that is kind of repetitive that is coming and going in Congo, it doesn't attract much more attention as it used to be then. That anytime you hear Ebola outbreak, yeah, that's voila. So that's how the world has moved. And it's important that we also reason in that direction. What made it so is because of the improved proactive and predictive genomic intelligence. However, Africa is still very much lagging behind. We are very much lagging behind. So, and what's the problem with this? In fact, recently, just May 2026, I'm coming. So, May 2026, the Africa CDC, Center for Disease Control or Prevention and Control. So they came up with this, I think back in 2020, 2019 rather, but there is a report that I'm also going to share with those who registers for this course, this webinar. So that they launched the Africa Pathogen Genomics Initiative, the PGI. I'm not sure we have anybody here that have heard of this particular initiative. So this initiative was set up in 2019 by African CDC. And despite the fact that we have 54 countries in Africa, only 46 were signatory or were participants in this particular initiative. So it started with 7 African countries, and Nigeria is part of it, of course. And what does this initiative, what does this have to do with what we are talking about? So we are talking about there is no nation, continent-wide collaboration or organization or survey or a work that put into place the genomics initiative that will help us to mitigate against new outbreak of diseases. So, and that's a problem if we do not have that preparedness. And that was why the Africa Pathogen Genomics Initiative was set up by the Africa CDC. And even though it's a lofty idea, Africa is still lagging behind, and it's a big problem. Although we are gradually, waking up to the challenges. So this systemic upgrade represents a massive step towards moving forward in molecular diagnostics and self-sufficiency and infectious outbreak resiliency across the continent, which is a very, very good news for us. However, we still have a long way to go, a very long way to go. And that is why we have to understand that, yes, we have a long way to go. It's also an opportunity for us to explore the challenges and make a name for ourselves. Uh, I'll try to. I think I still have it. I'll try to share the screen after this presentation on the communique that was released last month in May 2026 on this Africa PGI. So now the bioinformatics paradigm. So what bioinformatics does is it bridges science and computation. So we now have what we call the next generation sequencing. We have the computational phylogeny, and we have predictive epidemiology. In fact, one of the advances that we have nowadays, let me note it down so that I won't forget. Uh, Africa Center of Disease Control by Professor Christian Api. I'm going to refer back to that. So because it's important for this discourse. Now we have to bridge science and computation, and that is what bioinformatics is all about. So bioinformatics converts raw genetic data from DNA sequences into actionable public health insights. Like the information that you'll be getting is immediate, and you can immediately have a reaction or a answer to this particular response that you are getting from the genomic data. So it is the core engine behind that prediction. And what are those predictions? And we use next generation sequencing for that, computational phylogeny, and predictive epidemiology. Now, each of those steps are a very complex web, straightforward, and it's something that we need experts in. In next generation sequencing, you should be able to do fast mapping of complete pathogen genomes in hours. That means we need those who will be experts in molecular analysis, wet lab analysis. So we need experts in that. So after we've gotten results from all this laboratory equipment or laboratory procedures, we move on to computational phylogeny. And this is useful in tracing the precise evolutionary pathways of virus mutation. It is necessary for tracing the precise evolutionary pathways of virus mutation. And that is how we knew during the time of COVID-19 that we have Omicron strain, alpha strain of SARS-CoV. We have beta and the likes and the likes. So we use computational phylogeny generally, to trace the origin. Once we are able to trace the origin, we will know where to start the problem from. There is a particular strain of tuberculosis that was common in North Africa, and I didn't remember the year, maybe 2023 or 2022. There was an outbreak of tuberculosis in Spain. They have not seen that kind of strain already in Spain before. Like, yeah, if I'm not wrong, I think it's Spain or one European country. Now, the strain was so severe because it was not the people that had that strain, they were not responding to treatment. And oh, normal tuberculosis now use this, it will go. This thing they refused to go, and it caused a lot of public concern that they now had to start tracing the origin of that particular strain of tuberculosis. It was now later traced to North Africa, I think Morocco or Algeria, or with one country in North Africa. They now had to look for how they treated this particular strain of tuberculosis in that North African country. They now approached the treatment in Spain with that treatment regimen. So that's how it is very important for us to know phylogeny. If we're able to trace the evolutionary pathways, you'll be able to know which, how, and when to approach the treatment regimen for a particular disease. Now, after that, you can now do what we call predictive epidemiology, where you spot drug resistant strains before they spread. And this was what we did. For those who are students of our Africa Bioinformatics school, this is what TB Profiler, this is what it does. The tool used in doing that, it will spot drug resistant strains before they spread. You have the sequences. When you sequence it, you know which region or which strain is resistant to drug. You can also do that. You can also see that by phylogeny and sequence alignment. You will know this is there is a mutation in this particular location, and it's most likely an informative of something that's that is about to happen. So that's why this is very, very important. The bioinformatics paradigm is important for every one of us to understand. Now, there are different framework for predictive healthcare. We have three majorly. One is early detection. And I know Dr. Chooks will be interested in this. One, you are able to detect early viruses very early, like viruses like Mpox, Ebola, Lassa. For example, in Nigeria that has this always having this incidence of Lassa fever. I also read in a publication also that there are some other strain of Lassa. Some is it Lassa? I think it's one of those virus. I'm not sure, but I'm still going to read and give updates for those who are in our community. So even in Nigeria, I will remember the disease. I'm not sure, but I will remember the disease later on. Even in Nigeria, they have different strains and different seasons in which these strains do come up. And with that, it became a problem that we are not able to know, oh, which one is this, which one is this, which one is this. And with that, if you are not able to trace, it becomes a problem. Now, another example that I will give on this early detection was the case of COVID-19 also. COVID-19, everybody is predicting that it came from bats. Like some say it's from pangolin. Some said those are the two major sources that people that it was traced to, but majorly it was traced to bats because that was where the first incidents. The person that had the first incident of COVID was working in they were miners and they came in contact with bats in a cave and likes like that. So they traced it to bats. If we are not able to trace it to the hosts, the animal host, and this presents a problem, especially when your disease is a zoonotic disease. And what do we mean by zoonotic disease? They are diseases that are that transfer from animals to humans. So, if we are not able to trace it, that becomes another problem. So, this early detection is very important. So, early detection helps us to trace identify the virus and trace the animal host. Or if it is not animal host, maybe it's an environmental sewage samples. So, and then we know this before it escalates into the human population. Now, another aspect that I want to talk about in this regard is there was a study. There are different studies right now that are studying sewage samples. These sewage samples, they are testing it for COVID-19 or virus like that in the same class of COVID-19. Why? Because we saw that these virus, they can also survive in these sewage locations. And in that regard, it becomes a problem, especially in countries where they do not handle their sewage properly, which is a big problem on its own. So, routinely, researchers will take sewage samples and search for this potential virus. And if they are located, it is now easy for us to say, okay, this particular location, please, they should go and treat their sewage to prevent an outbreak of this particular disease. Now, number two is mutation tracking. This is another framework for predictive healthcare. We've been able to detect, but sometimes when our detection, we see this is common strain. Oh, this particular strain is common here, but we are not able to detect mutation. We are safe. But once we detect a mutation, that's another problem for us. Monitoring genetic variation over time to assess how changes might impact vaccine effectiveness or treatment performance. This is very, very important for us. Very important. Because right now, I know some of us, we exposed to different types of vaccine during COVID-19. Some took Moderna, some took different, I've forgotten their names, so different vaccine. Uh, so, in this regard, some reported that some vaccines are much more effective for a particular strain of COVID 19. So, which is another problem. These virus, their evolution, their mutation is kind of much more rapid, and that becomes a problem. We'll be able to trace the mutation and then apply the necessary health intervention. Number three is mitigation modeling. Now, because of what we have been able to collect, the environmental data, the health data, we can use algorithm to forecast mutation pathways and coordinate resource dispatch. So we use this for maybe through machine learning. There are predictive modeling, there are mitigation modeling that is done used in this regard. So we'll be able to mitigate that in these risk areas. For example, now one of the ways in which you do mitigation modeling is the case of mosquito. For example, if it were to be in the rainy season, we know that yes, this is the breeding time of mosquito. Now you can predict how rapid a mosquito or malaria via a disease spread by mosquito is going to be by the vegetation in that area, not just the season. You can also predict by the vegetation in that area, by the sewage in that area, by the so there are different factors that can assist you in modeling. So it is all this data you take together in order to make your intervention. No. So before we move to the next part, do we have anybody that wants to say one or two things before we move on to the second part? A contribution
Yeah,
or
thank you very much.
you're welcome, sir.
Just to say that I was in Nigeria recently in the Nigeria, the reference laboratory for the NCDC, and I
Wow.
met a group because that's where I was doing my analysis. And just like what you are saying, they are doing this wastewater monitoring, which they are using to monitor cholera, SARS-CoV, and meningitis.
Yes,
And
meningitis.
yes,
Thank you,
and those,
doctor. Meningitis is the one the disease I was trying to point out. Meningitis.
yeah. So
Thank
there's a
you.
lot of work going on when it comes to this issue of genomic surveillance, and it's a purely genomic surveillance work. I think it is being sponsored by Bill and Melinda
Mm.
Gates Foundation, and to support the NCDC for monitoring early detection of new diseases. I think this year, early this year or late last year, HG, which is now they've renamed it to they've renamed
H3
it.
virus.
Yes. I think they won Prosapi won $100 million grant for surveillance, early detection. Also, you can read it up for surveillance for new diseases. And recently, there's a paper published, a published paper about in Gombe. There was this case of a particular local government where patients were presenting typical of hemorrhagic fever, but every case was negative for they were all negative for Lassa, they were negative for everything, and they have to do some genomic sequence where they realized that it was caused by Peggy virus.
Hmm.
Or there's an association between, because 20 of the patients that died, I mean 21 or 21 of the patients that died, children, all had that Peggy virus. But Peggy virus is not known to be causing
Hmm.
death. That's hepatitis G. I think
Hmm.
G or H. You can read it up, Peggy virus also. But it was gotten through surveillance. And I think there's a reoccurrence of that outbreak again. So that paper and the findings is now um, being used to model a new policy for them, maybe a testing algorithm for that particular local government or response. If there's any outbreaks, they know that they have to test for Peggy virus. Thank you.
Thank you so much for the beautiful contribution. In fact, that meningitis and that you came up with, that you mentioned, was what brought me back. In fact, I knew about this meningitis of a thing and the strain in Nigeria while we were trying to apply for a particular grant. So, and I got to know about that. So, a lot of initiatives have been done in Nigeria. At least we know of Nigeria majorly, so that deals with all those things. So I hope we get there one day. Thank you so much, sir. So for the second part, Africa Pathogen Genomics Initiative. So there are different initiatives. So, but I'm just going to talk briefly on some of the initiatives so far and what have been done. So now the goal is to strengthen national public health institutes and establish sovereign data governance system. So there is this initiative by CDC, Africa CDC also, that is called Agari. Agari. I don't know why the picture did not show. So Agari, the goal is to keep Africa biological records within the continent. And it speaks to a problem that I do mention, that US has the NCBI, UK has EBI, Japan has DDBJ. Every country that I know that is serious has their own, they have their own database. They have their own database of this kind of data. So now Africa has its own Agari. It's called Agari by Africa CDC. So the initiative is to ensure complete regional governance and to bridge traditional silos and providing local researchers with access to data sets vital for domestic clinical innovation. So that's the major goal. And the comparison is given here. In the case of a traditional surveillance, it can take months, whereby genomic surveillance takes hours to days. And you can also predict. So the same thing is also in transmission tracking, drug resistance, and integration model. So because of this new data set, the new data houses that you store your data, so we now have it possible. And with the advent of genomic surveillance, it's much more rapid and faster and much more accurate. Now, it is noticed that yes, there is global genetic data inequality because it's kind of very expensive. It's relatively expensive. Only countries that are high income countries get this data in abundance. So Africa, because we are still kind of coming up, it's little, and the rest of the world takes 10%. So that's how far we have to go. So it's still a long way to go. And despite the fact that we are having the genetic diversity needed for that. So what is the roadmap for bioinformatics resiliency? Number one is scaling. So standardizing sequencing kits and NGS deployment to national public health institutes. Number two is training. And that is where NOBLEKINMAT comes to play. And government agencies cannot do it alone, because even in those government agencies, only a few handful has the ability. I will say it again, there is only few that you can point your fingers to in Nigeria that is doing what Professor Christian Happy group is doing. And the reason why the names keeps on coming is because they are doing big initiatives that's kind of very standardized. And we at NOBLEKINMAT is also looking forward to achieving that feat and also becoming more than that. So we have to train. We have to train people. H3 Bionet tried that. H3 Africa, H3 Bionet, they tried massively. In fact, they have open access courses that you can access that can train you in all this particular discipline. But it's not enough. It's not enough because we still have a long way to go. A long way to go. The population is still limited. And people do not see the right intense incentive to study all these things, or to go deeper in these things. Now, and that is another goal that we are focusing on at NOBLEKINMAT. So we are teaching people not just to learn the skill, but how to earn from the skill. Another problem now presents itself. Those who have the skills now, they are leaving Africa as well to pursue their own personal goal. And you can't blame them. The problem of brain drain, because what they have is not being appreciated back at home. And even if it is appreciated, the tools in which they will use to conduct those analyses are not available. For example, now if I want to sample a Sanger sequencing, I can do 100 samples within a week. As long as I can work. If I can do the extraction, 100 extraction today, I will send it for sequencing tomorrow and I will get the results by Friday. It's not like that in Nigeria. Before you even have the lab to sequence and send for sequencing, they will take your sequence, they will send it to South Africa. You have to wait before you get your data. That's how bad it is. Now, there is also the phase 3, which is connecting. We have to have a cloud network that cuts across different countries. It's very, very important. And that would enable us to do phase 4, which is predicting using artificial intelligence. Empowering Africans bioinformatics is they have local capacity deficit. And just like I said, NOBLEKINMAT bridges that gap between raw data set and real-time surveillance by training people. So we have training, we have diplomas, and we have consultancy services that we render to those who are interested. So, as it is said by Trevor Mundell of Bill and Melinda Gates Foundation, that pathogen sequencing will accelerate efforts to detect new epidemics before they spread widely and monitor their transmission in real time. This will contribute to better vaccine development and health security. So thank you so much. As we've gotten to the end of this presentation, I didn't even know we are going to spend up to 30 minutes, but this discussion keeps on getting more and more interesting. So if anybody has any question, so this is the right time to do so that we can have more chat and more discussion about that. Thank you so much.
Thank you very much. Please,
Yes, sir.
I want to ask a question. I'm just asking with regards to data policies and data privacies. And my question is, how safe is the African data when it comes to genomic sequencing? When actually we are using machine produced by other countries that host the storage of this data. For example, if you are using Illumina, the work I'm doing, I'm using NextSec 2000, and with the Illumina platform, NextSec 2000. Whatever we are doing, there's a back end, because when we are running, we had issues. We had to call the company. They viewed it and told us where the issue is. That means those data is not private. And I think in their policy, it is what it is. So how private are these data that is being sequenced using international platforms? That's one. Then two, my recent travel to Nigeria, and I saw that Japan has come to build a molecular lab, in short, a genomic lab within that place. The Jaga, is it Jaga or Juga? You can browse it up. It's a concept by the Japanese. They also have their machines and everything, which means that whatever we're also doing, just like we are calling the Illumina company, everything they are sequencing also goes to their own back end. And I'm believing that the guardian of this sequence or genetic data is the new, good mind of tomorrow. When it comes to control, comes to human inventions and human monopolies, so as to it. So I just want to be clear: how safe, how private are these genomic data that we sequence using international platforms?
Those are interesting questions, actually. And actually, the general problem in terms of data proprietary is not unique to Africa alone. It's a global phenomenon. And even though the severity it's kind of more than uh, uh, each other, but I know for a fact that each of these companies they always put all this backdoor. Apart from even from genomic sequencing platforms to even a military installment, so they have all this backdoor. If they sell a military item to you, just forget it that they have a backdoor into that software or that tool. So having understood that problem, so I think it's also a wake up call to African leaders. But I don't think we are. I don't think we are there yet. So it's actually a problem that we have to deal with, at least for the time, the foreseeable future. And even though it's not peculiar to Africa alone, so it's also rampant across the globe. But yes, the scope at which they harvest that data, I know for a fact that in Africa, there is kind of unrestricted harvesting. They will just be harvesting the data, harvesting the data. So, but in Europe and other places, I know there might be some limitations to kind of data you can get. So, for me, I think it's just for us to keep working, and when we get there, God has always find a way to make us survive all those kind of tsunamis, and it is what it is. Let me just put it that way. It is what it is. Professor, you are raising up your answer.
Okay, okay, thank you very much, Dr. Ashraf,
Good
for
evening,
the
sir.
enlightenment. Yeah, yeah.
Good evening, sir.
Ah, ah,
I just allow me to speak. I
Good
beg.
evening, sir.
Good evening. Who is great?
How
Professor,
are you?
Uh,
Professor new.
thank you very much,
Hello.
Dr. Ashraf. Hello, hello. Can you hear me?
Professor Adeni, yes, you can go on, sir.
I said sorry I joined
Okay,
lately.
you are welcome, sir.
No problem, sir. You're welcome, sir.
I want to apologize for joining lately because the topics or the line of discussion that I met was something that interesting me too because currently I'm just preparing samples to be sent to the US with my collaborator, and the samples are malaria parasites, and we are using genomics approach to
Unravel.
sequence malaria protein and to unravel the diversity study. And the challenge is with all of these samples that we are sending, Then who owns the data? We are transferring African data to American soil. And then the question of or the challenge of the ethical issues or even the ownership of the data. Because once you transfer it to them, you can't claim
Take it
that
back.
you own it again.
Yes,
Eh?
yes.
So, and because after these samples are sequenced, it will be deposited in their NCBI or wherever, and then. But to me, the problem is with us, and the problem with us is that we should also upgrade our facilities too, even research centers, universities, so that all of these things we should be able to do it at our own level rather than sending it to a strange land where the ownership becomes a real challenge and difficult for us because and Africa has come a long way. Because if you look at it very well, for instance, in the case of malaria parasite now,
Hmm.
those people, there's no malaria, even if they have mosquitoes in the US or even in the UK. It is the sub-Saharan Africa that has a larger percentage of malaria cases. And so there's nothing serious that they can do from their end without including researchers from African countries, especially West Africa, and to make what they are doing to make people
It's
recognize
meaningful.
or see meaning in what they are doing. So what these people want to do is we are developing a kit that will detect malaria parasites within 2 minutes, and we we require no electricity. We we cost less, as in somebody put a question that prof, $2 is too much, because $2 if you multiply it with Nigeria, at least around close to 3,000 naira. If you want to detect malaria parasite in me, and you are asking me to pay
Pay
3,000 naira.
3,000.
3,000, that 3,000 naira can buy anti-malaria now, and I just, whether or not I have malaria, once I take anti-malaria, then I know that I'm free, sort of. But I guess at some point we have to sit down and take a review of this price is too much, and then hospitals may not be able to afford, because we will still do clinical trials for like 2 years in Nigeria, and before that, then we can start to think of how
Going
the price
commercial.
that will be the affordable price. Although that price may not be a big issue for researchers like us, but for a layman who has malaria and need to know whether malaria parasite is present anymore, it might be a difficult thing, because we are poor in our country. We are not rich. We cannot hide the fact that we are poor.
I
So
will not accept that we are poor.
we are poor. Are we not poor? If you look at
No, we are not poor. No, we are not
ah
poor.
so okay, if you go to the north, you know that we are poor. Nigeria as a nation is rich, but the
Yeah.
population, the people there are poor. Nigeria as a nation is rich, but the people that are living there, majority are poor. So that's the truth. Nigeria is rich as a nation, but when it comes to people that are there, they are poor. So the blame pushed back to government that the wealth is not common, and they are deceiving us as commonwealth. So God will help us.
Amen.
God will help us.
Thank you so
So
much, Prof.
that story is that line of discussion interests me so much, and I was happy
Yes,
that it's being
it was
raised
brought up.
by you. Yeah, it was brought up by one
Doctor.
of our yes.
All right. Thank you so much, sir.
Dr. Chuks,
It's
thank you for bringing it up.
Thank you, Prof. Thank you, Prof.
I appreciate you for bringing it up, because it shows that you love Nigeria, and then you are pitying us for what is happening, and then we should look inward to save our country. Almighty God will help us.
Yeah, Prof, I'm still part. I'm not pitying. I'm fully in. My neck is in there. My research work. No, my research work is actually in Nigeria.
Okay.
Last year I was in Nigeria 4 times, so I spent half of the year in Nigeria. This year I've been
Wow.
there. I'm going to come back again. But the issue is this, Prof. I didn't want to say it because I don't know those are on this platform. But let me just give you a glimpse of what happened during my research.
Wow.
We're talking about ownership of data. We're talking about, just for us to host data, yeah, and issue of material transfer, MTAs, material transfer agreement can be there for the ownership of data, that the data can be sequenced or analyzed in so and so country, but it belongs to, so a proprietary act can be added to it or clause in the methodology, and you know that it belongs to you. But that's just like a name that is belonging to you. I'll give you an example. We had the work I'm doing, one of the works I'm doing is one of the most funded work from the UK government that was
Wow.
planned in 2022 downwards, because they felt that mpox was going to be a global, more serious thing, and actually they were disappointed when it didn't go as global as it got here. And because the sample size and everything was, you see the
It's
being
actually funny.
yeah, but during the plan and the method, during the plan of the research, Nigeria insisted that they are going to host the data. And
Wow.
I'm not going to call the names of the area so the money was given, I believe, for hosting in Nigeria soil. But Prof, one morning we were trying to enter the website, we couldn't enter. I would loaded that place with a lot of work.
What happened?
What happened? And what happened is the question. They were trying, according to them, they started speaking many English that we at Oxford couldn't understand. They were trying to upgrade the server, and they did not back up.
Hey.
Ah,
Hey.
That's what watered down my research work. It watered it down that they said this work cannot pass for a PhD. I have to quickly, thank God I'm right. I wrote on that proposal to add to it. The money was given. So when we talk about not being ready, there's a lot of things. There's a lot of things. Okay, now we are done with the work. We are trying to, settle down, do one or two things. And we are not seeing them. When you are not seeing them, later you say you want to be first author. When you didn't do analysis, you didn't write up, you didn't do the data cleaning and the rest. How do you still this is within the context of our own. As we get our mentors, mentees, we need to really speak to ourselves. But there are simple things that could help. You're talking about even taking the data to be analyzed somewhere else. I know that material transfer agreement must have been signed and the rest. Even this genomic class we are having, that we are analyzing our data through Galaxy. If I put in all my data in Galaxy and do the analysis, how private is that data? It's no longer
exactly.
because the back end is being seen by somebody. It's not it's not automated. There are people, even if it's automated, there are people that controls garbage in, garbage out. There are some people that controls the input and the output. There are people that can interpret it. There are people that are doing monitoring and evaluation. There are people that are actually seeing how to improve the work. Therefore, they are seeing everything you are doing. So data proprietariness and privacy is even something else that ethical issues need to look at it critically and come to an agreement. But any agreement that come in is just going to be on paper, because the real act still remains that he that blows the piper will determine the tone of the dance. That's the truth.
Thank you so much, Doctor. Professor Adeni.
I'm enjoying the presentation.
I want you to also, I think one other person also wants to contribute, but
Yes,
before
okay. Thank you very much. All this is very interesting. That's why, because the aspect I just want to talk about is also key factors affecting this our home Nigeria. So when you talk about are we really ready, I think the answer for now will still be no. If you itemize our institution in terms of our molecular readiness, you see that it's very, very, very low. And so which implies most of the, strong molecular analysis we do is either you send to South Africa or elsewhere, depending on the capacity and the collaboration. So that's one. Two, who sponsored most of the research with higher, molecular data? It's not Nigeria. So the question is, are we even that's it, because you are talking about ownership. Who even sponsor it? The key one, the high one, all the COVID-19 analysis. Are we the real sponsor? That's the question we
Because they
continue
will
to
claim
ask
they will,
ourselves.
they will claim the ownership as well because they were the one that paid.
Yes.
Yeah.
Like for instance, since 2018, we have been doing insecticide resistance monitoring in malaria vector, and this is being sponsored by Global Fund. So they just use NMAP and NIMA as a base, as like they are just how will I call it? They are just
Like
collaborator based.
placeholders.
Yes.
Digerewu.
So most of so that's my main worry that prompts this question. I want to ask the expert on this platform and the host, Dr. Ashraf, that this thief, there's one aspect that is very important. Predictive intelligence model. Yeah. Now, if you don't have the data, how do you become expert in that aspect? Even when you have the data, you are not the owner. So the question is, can we have a kind of especially for early researchers, can we have a kind of I would like, or maybe, if you call it framework of how can it become an expert in that? We putting in mind all these itemized problems affecting
Challenges.
Africa, especially Nigeria. So that's my, because I think other ones has been available because that one will really help Africa in most of these. Today you have this outbreak, tomorrow you have this outbreak. So this problem, I know we have not solved it, and then just as the prof and the other colleagues have been itemizing them. So if we are to be listing this problem, I don't think we can finish it. So let's assume this problem are there. This is where we are, and we still want to move forward. So especially to be relevant in solving our own base problem. So can we have a kind of a guideline towards achieving such a predictive, or as you call it, proactive predictive intelligence? That's the aspect I'm trying to ask based on what we have.
Thank you so much, Prof, for the question. I actually, these are projects that not even an institution can solve in terms of the human resources and the capital resources that it will require. I know for a fact that Africa Biogenome Project that is being led by Dr. Ebenezer Thank God, also in UK. So it was born out of this kind of necessity as well. That yes, we have the flora, we have the fauna in Africa, but we are not sequencing our own data. So, and he was able to, it's a very big work. It's hard work. It's unrewarding. Let me put it that way. Only that you know that you are building a legacy for yourself and for Africa, and that people will remember you for good. So, and so he did it, and he started the work. And the reason why he was able to start the work was because he's already belly full. Like he's not hungry. So, but he has time to think outside the box and he knows where he can go to get the resources to do that. And when he started, he encountered a lot of challenges. And if not because of the passion and the dedication he has, he might not be able to get through it. Even despite the success of this Africa Biogenome project that we are seeing small, like little by little, there's still a lot yet to be covered. In fact, they just sequenced Agbaluma, Africa walnuts. They just sequenced the genome. They just picked different types of plants. So they just sequenced the genome, I think, early this year or late last year. So these are initiatives that individuals are coming up and putting, like solving a solution. I also have the reason why I made mention of that of Africa Biogenome project was because I have an idea of how to go regarding solving this problem of creating a database of different resources. He focused on the full genome. My initiative, I'm thinking, okay, let's not even go far. Let's start with the Sanger sequencing, which is the barcode. There's a name I give to it. I'm still going to when I have time. When I have time, and I know that there are one or two people I can call or reach out to assist in the project. I know, but it's something that will require a lot of time and effort. So these are things that individuals can do, and then we can invite global partners to join. Now, to your question itself, I think one, there is no coordination or cohesion in terms of coordinating all these initiatives. And actually that's just the sad reality. Um and the reason why I'm saying this is because not because I that's just the reality. Don't let me sugarcoat or say anything to make dress it up. Let me give you an example. There are three main let me just say four main bioinformatics company that I know that in Nigeria. NOBLEKINMAT Limited, Arc Bio, Elix Biogen, Genomic. Those four, like at least I know, at least if not any other person, I know NOBLEKINMAT started before most of this initiative because before they became more popular. But we have our own challenges, and there is no, I'm saying this because not because it's an outsider information or something like that. I'm saying it because it's something that concerns me. I know there is no synergy to coordinate the efforts of individual companies like that. Yes, there are some. I know the founders of all these other companies. At least I've spoken and I've been invited for ArcBio webinar before. I spoke on ArcBio. I've spoken on platforms belonging to Genomark. It's only on Elix that I've not been invited. And it had its own reasons too. According to some, like, everybody's trying to make a name for themselves. Let me just put it that way. Everybody wants to be at the forefront that yes, I'm the champion of this, I'm the champion of that, and that's a big problem. And that's a big problem, especially for we Africans. Like, it is peculiar to Nigeria in particular. Everybody just wants to be, yes, I am the champion, I'm the champion, I'm the champion of this. We don't want to give sincerely to an initiative that doesn't carry our name, that doesn't carry our identity, that doesn't like. But even though that initiative that thing is doing is kind of for the greater good, we might not want to contribute to that, and that's a problem. It's, I would say it's human problem, but it is much more of a problem, especially regarding Africa and Nigeria. And now I'm just talking about the private owners of bioinformatics companies. Now, this is now the benefit. With the advent of all these different companies, we can even have more than that. Yes, it will create a kind of competition for a market that is still nascent. It's still growing. Like, no, before when we first started bioinformatics at NOBLEKINMAT, at least I remember very well in 2019. We first had to tell people what bioinformatics is. Bioinformatics is this. Bioinformatics. in that way we call it the blue ocean strategy. No, the I think green or red ocean strategy, something ocean strategy. You first have to create the awareness for the need of the in the market before you now start marketing that thing to people that you need this. Like now, for example, now if you want to stage any course in Africa on bioinformatics, you are deceiving yourself if you are putting the price at no, or if you are putting the price more than 100,000 naira, you are deceiving yourself. Nobody will buy. Like if people will buy, it is people that know they need it, that they will I need this thing, no, I need this thing, no, I need this thing, no. That's when they will buy it. But you know that a lot of people need this skill, but you first of all have to educate that bioinformatics is this, is the next gold mine, is it so it is not okay, okay, oh yeah, let us come and learn now. Oh yeah, you have to pay. Oh yeah, come and pay. How much? I don't get money. I know they do again. So they go. So that's another big problem for bioinformatics, especially in Africa. So the problem is hydra headed. As you are approaching it from this angle, you have another problem here. You have another problem here. But as I said, it's just the right step in the right direction. We are creating the right awareness, and people are getting to adapt to it and adopt it. I can remember when we first did our first campus tour in Nigeria. Professor Yola accommodated us in University of Ilorin. I had the luxury of meeting another passionate individual in Fountain University, a lecturer. She spent her money to invite me. Professor Yola also spent his money to invite us to Ilorin. So we went to Crescent University too, by Professor Suleiman, also accommodated us in Crescent. We also went to Babcock. Babcock, in fact, was so grand that they spent money, and even though we spent one day there, so that's to show that people are gradually adopting and adopting. But the clearer picture is still yet to be done. So if we are going to bring an initiative that would make more meaning or can be interpreted into more impact, we first have to create the awareness that everybody will see that yes, we have a problem. Oh, we have a problem. When they understand that, then the initiative can come in. So other than that, we might just be wasting our time. But just like you said, we still have a long way to go, and I'm confident that yes, we are moving towards the right direction, and even at a speed that it's even impressive to me as well. So
Sorry,
I hope we get
let me cut him because
yes, doctor.
if we start analyzing the problem, we will spend the whole day. But I think one of the way forward is yes, you are trying to utilize the available media. I got to know about this through LinkedIn.
Yes.
And the position where I occupy now in Oxford, I got it through LinkedIn.
Wow.
But that is not enough. We have two profs here, and they must have grown through the rank. They must have been HODs or deans in the faculty. One of the way to go, which I'm suggesting, which I'm trying to work hard and we'll see that we'll get it through, is for you to first of all institutionalize what you are doing.
Yeah.
And this can only come through collaboration. Noble marks can remain this way in the next 30 years if we don't institutionalize it. And how do you do that? I have a mental picture of what could happen with Noble marks if we work together and get institutionalized. The first thing to do is, I'm a consultant public health physician. I have my I did my residency 6 years and fellow of the National Program Medical College of Nigeria, Public Health and Community Medicine. And in that body, I was a trainer before I left, but we have curriculum review committee. One of the things you can do is to see, we can begin with low-hanging fruits. It could be seminars in these two profile here. They're interested in bioinformatics. By now, they might not be able to pursue it passionately the way we are, younger generation. It's for them to
Yes.
identify younger generation they want to mentor and say, see, there's a future
The
going.
profs are young, go. They are young, go. They'll say,
No, they're already professor of something. They said they want
yes,
to be
yes,
double
yes.
prof.
I'm
So,
just joking.
so
I'm
we
just
have
joking.
mentors or mentee they want to pick up and say, see, I want to direct you,
Yes.
go in for bioinformatics, and
Hmm.
I will see how to guide you.
Hmm.
These institutions, if handled by departments or particular body, can begin to award diploma as well as diploma, especially when it's tied to university, diploma in
Yes.
bioinformatics, having their school name.
Yes. Yes.
So you are selling a product
Hmm.
to a school, to a university, to start up a department or a program that they will
Hmm.
benefit from, and you are bringing the manpower.
Hmm.
With that, if you write your proposal well, they will agree.
Yes.
And I have professors, I have people I can send to you today. We'll have a meeting, we'll collaborate, and they will tell you how to penetrate, if it's National Postgraduate Medical College or penetrate the review committee or public health. They are there. Why they will not run with it is that they don't have the capacity.
Hmm.
You be in the class, you are talking about German, Seman, all those languages that to us is a new ground. We are learning everything again. It's like we are going through the ABCD. We are getting frustrated in some commands. We didn't do arrow while in school. Now I have to start learning arrow. I need to start learning bioinformatics when I thought I've reached the peak of my career. And I'm starting all over again. But
Yeah.
if we know that there's an available platform where we can reach people, it's collaboration. A diploma, even diploma is high, certificate of certifications of this. Before you know, 2-3 people will be interested. And in 2010, I first met Professor App in 2009.
Wow.
When I came back from youth service and started residency in 2010.
He was still in UI that time.
Yeah, but he just finished from Harvard and he was coming from
Yeah,
Harvard.
he came back.
He had this
Postdoc.
collaboration with Harvard and he was working for Lassa, working on Lassa fever. And what was the issue? Professor Api kept insisting that he wants African-based scientists.
Yeah.
All the collaborations, he would tell them it must be situated in Africa. He kept saying that. Yeah, it is Harvard, but so then we were calling Professor Api Harvard because I was in Iro Specialty Hospital then, and we're having two institutions that were collaborating, Harvard University and the Germans. So in that place, we have two buildings. We call this one the German and the Harvard. But he kept insisting that Africans, scientists by African must be raised. And today, he changed his name from AGID now to IGH. IGH.
Okay.
So please, let's see how we can work together to institutionalize what you are doing. Once that is institutionalized, getting collaboration, because you cannot get grant easily through this way. But if investor for Abuja public health department is talking about bioinformatics, which you are a major collaborator, then you can know that okay, we are applying for this grant through investor of Abuja. Yet you know you are the primary person at the background, or investor of Ilorin. Or so collaborations, institutionalizations, and all this is one of the ways to go. And because it is true, institutions, Bill Gates or Melinda Gates Foundation will not give you money to use those machines. This year, happy I have the LinkedIn this in $100 million grant.
That's massive.
Yeah, it's massive. And that is why every time you see him, that is among the 100
most influential researchers in the world.
And when you get to HG, they actually head there, the machines there, you know, you are in another place.
Exactly.
Redeemers University. So what did he do? He went to pitch his niche with Redeemers, a private university. He couldn't have done it himself as happy. He could only do it through university. So he built an institution. That institution is his institution, but he bears the name of Redeemers University. Collaboration, institutionalization.
Collaboration with wisdom.
Thank you. That's
exactly.
what I'm saying. But we can talk over this
Definitely.
again and again, and we'll draw the roadmap. And you see, before the next year, you see that you've gotten what you wanted. I have a recent person that started another group now, whether it's taking over. I met him in NCDC. He's schooled in UK, came over. What's his name now? If I go through my LinkedIn now, I'll get him. But they're already pushing. I will look for that LinkedIn post and send to you and see how this prof is moving already. He went to institutionalize this NGO, and that's what's
That's interesting.
All right, thank you.
Thank you so much. Thank you so much, Doctor. In fact, today is an unexpected kind of interesting interactions that it's random. In fact, I never expected this so to be in depth, and I so much appreciate. I know those that missed today's session, they don't know the value of what they've missed so far. So, and I so much appreciate the contributions of everyone, and surely the two profs here alongside me and Dr. Chooks, we are still going to meet and discuss in depth on way forward on how to go.
Thank you. As if you know what I have in mind. Thank you so much. We have to.
So,
we cannot stop here.
Yeah, definitely.
Yeah, we have to continue the discussion.
Actually, I would maybe we'll fix a time so that we can have more in-depth meeting. And then we can discuss on the strategy.
And
So
also
thank you so much.
Proposal. Think of
Yes,
the proposal
definitely.
for Africa.
Yeah,
it makes sense.
Thank you so much, sirs, and it's a pleasure to have you here. And till our next monthly webinar. So I can't wait to see everyone. The recording will be provided through the links. We've had the recording of session, so if you miss any parts, you can catch up on what you've missed so far. So thank you so much, and see you on the next one. And have a nice day ahead. Bye.
Thank you so much.
Thank you, everyone. Dr. Chooks,
Thank you.
thank you.